Breast cancer is a mass of mutated cells that grows uncontrollably and continues to multiply, forming tumors that can be benign or cancerous. If left alone, the tumors can continue to spread throughout the body. These symptoms appear as lumps in the breast tissue, pain in the nipple or breast, swelling, and more. The types of breast cancer are divided into categories based on the proteins they impact. The three most important proteins affected are the estrogen receptor, the progesterone receptor, and the human epidermal growth factor receptor 2. Celebrities like Angelina Jolie and Olivia Newton-John are bringing awareness to breast cancer, and with the disease developing in 1 in 8 women in the United States, it begs the question, should we all be doing genetic testing for breast cancer? Only 5 to 10% of breast cancers are hereditary, the most common mutations happening in the BRCA1 and BRCA2 genes. Having a family member with these mutations does not guarantee that the rest of the family will. Mutations in the BRCA1 and BRCA2 genes cause roughly 10% of all breast cancers, and when combined with SNPs in other parts of the chromosome, the risk of having breast cancer increases. Women with BRCA1 and BRCA2 mutations who have breast cancer typically have a family history of other cancers, such as ovarian cancer. Women who have these mutations have as high as a 72% risk of having breast cancer, and mutations in BRCA1 and BRCA2 also increase the chances of ovarian cancer, colon cancer, and even pancreatic cancer. Some new gene mutations are associated with breast cancer, but none appear to have as dramatic an effect as BRCA1 and BRCA2. Mutations in the PALB2, PTEN, and TP53 genes are defined as high risk, meaning the chances of having cancer sometime in life is over 50%. Mutations in the ATM and CDH1 gene are at a moderate risk, anywhere between 25 and 50%. Other genes that are still being researched but do not have sufficient data to form its conclusion include the BARD1, BRIP1, MLH1, MSH2, MSH6, PMS2, EPCAM, RAD51C, and RAD51D genes.
Okay, so Sam, do you want to record hers? And then it's a case of...
A way to identify breast cancer mutations is through genetic testing. 23andMe has become one of the most popular ways to perform genetic testing for the general population. We gathered some statistics for the 23andMe testing of breast cancer. There are numerous SNPs tested by 23andMe that are related to increased risk of breast cancer. SNP RS2981582 and the FGFR2 gene was one of the 4 strongest associations found in a genome-wide association study of over 4,000 breast cancer samples. The FGFR2 gene makes a protein that is involved in numerous cellular and bodily functions, including cell division, bone growth, and development. A mutation in the gene caused by the RS2981582 SNP can cause the FGFR2 protein to become overactive, which would lead to cancer. A woman's chances of developing breast cancer normally are 1 in 8, but a FGFR2 variation would lead to a 5 to 10% increase in risk of developing breast cancer. Another SNP that has been shown to increase the risk of breast cancer is the RS3803662 SNP. It's located in the TOX3 gene, which is a gene that encodes for a protein that is involved in multiple cellular processes, including apoptosis, which is one of the ways that the body gets rid of unneeded cells. The variation of the TOX3 gene caused by the RS3803662 SNP blocks the apoptosis from occurring and leads to cancerous cells. One study found that the risk of breast cancer increased by around 24% for those affected by this mutation.
23andMe fails to recognize the importance of the hereditary breast and ovarian cancer syndrome known as. In this variant, a deletion occurs in the gene and everything else shifts over. This affects how the codons are read, affecting amino acids and protein function. According to the HACCP journal, this variant is highly frequent and has affected up to 30 families. The typical function of the protein is to protect one from getting certain types of cancer, and when the deletion occurs in BRCA1, one is more prone to getting cancer, more specifically in the alveolar and ductal epithelial cells of the mammary glands. Since this variant is highly frequent, one's chances of having it is very high.
Although there are many positives to receiving a genetic test for breast cancer, there are also arguments against the notion. Some of these arguments include treatment before the actual symptoms of the disease and other ethical problems such as families being disrupted. Testing negative for gene changes that increase the risk of breast cancer may bring on feelings of guilt, especially if other family members do carry the gene changes and face an increased cancer risk. Another concern is that a negative test result doesn't mean you don't have a risk of cancer. Genetic testing is moving from targeted investigations of monogenetic diseases to broader testing that may provide more information. However, there may be ethical reasons to not screen for breast cancer variants in a panel or to revise how testing and disclosing of results are carried out. BRCA1 and 2 testing, or multiple gene panel testing, is usually initiated by the individual for two typical reasons: by an individual who is concerned with the number of cases of cancer in the family, or by an individual with a form of pre-existing cancer to provide clinical guidance for this particular case. Once a pathological variant has been detected, this generates a risk profile, not only for this person, but also for his or her relatives. A genetic test can be initiated by someone who worries about his or her family history and is motivated by a desire for reassurance that they themselves are not at an increased risk. Because relatives may be approached after a positive genetic test, cascade screening, which specifically helps identify hereditary diseases, is an option. The distinction between testing and screening has an ethical importance from the point of view of autonomy. Usually, the individual seeks healthcare to have a problem remedied. This is at the initiative of the person themselves. It is important that all follow-up testing and treatment are decided upon in a dialogue with the patient according to their needs and desires. These tests also come with limitations since they cannot provide every detail about the disease and can come back inconclusive.